I'm going to repeat again this. The first step is the removal of the amino group, as I said. The amino group will be translocated to alpha ketoglutarate, and the amino group is converted to alpha keto acid. This is called the carbon skeleton of the amino acid. The alpha keto acid of alanine is pyruvic acid. This is called the carbon skeleton, means the hydrocarbon without the amino group. It is alpha keto acid. This group is called keto. This is a keto group. And alpha ketoglutarate accept the amino group from all amino acids. It is converted into glutamic. That's why I said glutamic is a boss or a president, because it accepts the amino group from all amino acids. This enzyme is called ALT. AL means alanine. T means transaminase. Trans means transport. Aminase, transport amino group. The second enzyme is AST. This is a spelling mistake. AST. SCT. AS means aspartic acid. T means transaminase. The amino group of aspartic is replaced by alpha keto acid. And this is a carbon skeleton of aspartic, which is oxaloacetic. The amino group go to alpha ketoglutarate. Alpha ketoglutarate accepts the amino group from aspartic and it is converted into glutamic. What is the clinical value of the plasma transaminases? Aminotransferase is an old name. The recent name is transaminases. Trans means transport. Aminase means transport or translocate amino group. These enzymes are normally found inside the cell, intracellular, and they are mostly found in the liver cells. They are also present in the muscle and the cardiac tissue, and all of these enzymes are intracellular, so they are not found in the plasma. If there is damage of these cells, if the liver cell is damaged by a virus, such as hepatitis virus, damaging the liver cell will lead to the release of these transaminases from inside the cell. It will go to the bloodstream. So AST, ALT, okay, are of diagnostic value. They are elevated. The level is elevated in the blood in all forms of liver disease, such as viral hepatitis or toxic liver injury, if the liver is exposed to toxins. These enzymes are also elevated in non-hepatic, yani non-liver disease. Non-liver disease, such as damage of the cardiac muscles, myocardial infarction, or damage of the muscle cell. Muscle disorders means muscle diseases. If there is a damage of the skeletal muscle as a result of disease, or damage of a cardiac muscle as a result of infarction. Infarction means thrombus, which occludes, blocks the coronary artery, so the cardiac muscle will die. What are the enzymes which remove amino group other than transaminases? We have oxidases. Oxidases. L-amino acid oxidase. This is amino acids. What's an alpha carbon? Alpha carbon is the carbon atom. This is an alpha carbon, which is the first neighbor to the carboxyl group. This is the carboxyl group. COOH. A chemical group, functional chemical group called carboxyl group, which is unique to the organic acids. The first carbon atom, which is the first neighbor to the carboxyl, is the alpha carbon. We have amino group in the alpha carbon, so this is called alpha amino acid. If you have amino group in the beta carbon, this is also amino acid, but it is not found in the protein. In the proteins of the living organisms, the amino group should be present in the alpha carbon. We may have another amino group in the beta carbon, in the gamma carbon, but if you don't find the amino group in the alpha carbon, this amino acid is not found in proteins, okay? What is an oxidase? An oxidase is an enzyme which catalyzes oxidation, means the removal of hydrogen. We remove 2 hydrogen atoms and we give it to a carrier. The carrier, hydrogen carrier, FMN, is a nucleotide of riboflavin, which is vitamin B2. This carrier, which is vitamin B2, will carry 2 hydrogen atoms. Where does the hydrogen atom go? It will meet oxygen. It will meet molecular oxygen and leads to the formation of hydrogen peroxide. Hydrogen peroxide is toxic to the cell. This is a free radical, one of the reactive oxygen species. That is why I said excess protein is dangerous to our body. Again, oxidase, enzyme which catalyzes oxidation, removes 2 hydrogen. This is the first hydrogen from here, the second hydrogen from here. So we get C double bond NH. This amino acid is unstable. It will be broken spontaneously to give keto acid. Why it is alpha? Because this is alpha carbon and this is a ketone group. The keto acid is the carbon skeleton of amino acids. L-amino acid oxidase has low activity, has very weak activity. It is found only in the liver and kidney. Again, 2 hydrogen atoms are removed. They are accepted by a hydrogen carrier, which is FMN, riboflavin nucleotide. And this hydrogen, which is carried on FMN, it will meet molecular oxygen, react to the oxygen to form hydrogen peroxide. And we will study how hydrogen peroxide is removed from our body by an enzyme called catalase, which convert hydrogen peroxide into 2 molecules of water. Alpha amino acid is not important to memorize it, but you should know that alpha amino acid is converted into alpha keto acid, which is the carbon skeleton of amino acid. The second enzyme, very important, glutamate dehydrogenases, and it is L also. L-glutamate dehydrogenases. Glutamate is glutamic acid. It contains the amino group in the alpha carbon. This is the carboxyl. The first neighbor is the alpha carbon. So glutamic acid has amino group. I said glutamic acid accept amino group from all other amino acids, from 19 amino acids, and from all amino acid which is not found in proteins, from all other non-protein amino acids. Glutamic acid undergoes oxidative deamination. Oxidation means removal of 2 hydrogen. 2 hydrogen are carried on NAD, but it is NADP. At this stage, it is NADP, niacin vitamin B3, which contains phosphoric acid. Do you hear me? Do you hear, or the internet unstable? Do you hear me? Or you are muted? Do you hear me?
Yes, we're hearing.
I want to make sure that you hear me in order to complete.
We're hearing.
Yes, yes, okay. Removal of the 2 hydrogen atom and removal of the ammonia. Of course, this is not a single reaction. This is 2 reactions, but they are abbreviated in a single reactions. Removal of 2 hydrogen, and they are carried by NAD, niacin, vitamin B3, but here it is NADP, NAD which contains phosphoric acid. Okay, and there is also removal of ammonia, and glutamic is converted to ketoglutaric. And we have studied alpha ketoglutaric in citric acid cycle. So amino group is replaced by ketone, which is alpha carbon. Ketone in the alpha carbon, alpha ketoglutaric. And then we are going to study where does amino group is released as ammonia, NH3. and it has a certain metabolic pathway to get rid of this ammonia. This process is called transamination. With oxidative deamination, it is called transdeamination. Means two reactions, transamination and oxidative deamination. Again, this is a transamination, transfer of the amino group. Amino group is transferred to alpha ketoglutaric, and we get keto acid. This is transamination, transfer of amino group. The second is oxidative deamination, which is oxidation with removal of the amino group. Oxidation, as I said, 2 hydrogen atom is removed on a carrier, and then amino group is liberated in the form of ammonia. So this is oxidation with deamination. Again, glutamate dehydrogenase, there is oxidation, removal of 2 hydrogen, which is carried on the NAD, and deamination, removal of the amino group in the form of ammonia. Okay? And then we get alpha ketoglutaric. Combination of both is called trans, means transamination, and then deamination means oxidative deamination. This is a combination of two reactions, transamination or aminotransferase. Amino group is removed from amino acid and it is converted into keto acid. All amino acid gives the amino group to ketoglutaric, and ketoglutaric is converted to glutamic. So this is a boss or a president, because it accepts the amino group from all other amino acids, and then glutamic undergo oxidation and deamination. Oxidation means removal of the hydrogen from glutamic. Deamination, removal of amino group in the form of ammonia from glutamic, and glutamic is regenerated, converted into alpha ketoglutaric. Is everything clear? Is everything clear or not? Please answer. Everything clear? Anyone want to ask me any question? Please answer me. Respond to me, please. The internet unstable? Is everything clear up till now and the internet is good? Okay, the last slide. I suggest not to study the last slide now, because it will make some confusion, because it looks confusing. I suggest to study transamination, oxidative deamination, and combination of both, trans-deamination, and then to study the last slide. I suggest to give the last slide tomorrow, because this is our specific method. This is not transamination, and this is not oxidative deamination. This is a special method for removal of amino group from a specific amino acids. Okay? So I suggest to leave this slide to Saturday, tomorrow. Okay? I suggest. Anyone want to ask me any question? Okay, I will repeat transdeamination again. Okay. This is transamination. Trans means transport, translocation. I translocate the amino group and give it to alpha ketoglutaric. This is keto acid. The amino group is translocated and it is accepted by this keto acid. This keto acid is converted to glutamic acid. Okay? And this amino acid alanine, the amino group is converted into alpha ketonic oxygen or keto acid. The keto acid of alanine is pyruvic. This is called carbon skeleton. Carbon skeleton of the amino acid. Again, the amino group is accepted by ketoglutaric acid, translocated, is given to alpha ketoglutaric, which is converted to glutamic. And we have studied alpha ketoglutaric and glutamic in the citric acid cycle. This amino group will be replaced by ketonic oxygen, by ketone. This is called keto acid, or the carbon skeleton of alanine, which is pyruvic acid. So alanine, when you remove the amino group from alanine, it is converted into pyruvic acid. Okay? This enzyme is transaminase or aminotransferase. Another enzyme, AST, this is not A, this is S. This is a spelling mistake. Aspartic acid is an amino acid which contains amino group. We need to remove the amino group. We remove the amino group and replace it by ketone. The keto acid of aspartic is the oxaloacetic. So oxaloacetic is the carbon skeleton of aspartic acid. Aspartic is an amino acid. The amino group is accepted by ketoglutaric or translocated to ketoglutaric to give glutamic. So this is transamination. I will stop here. Do you understand the transamination? Okay, I will answer this question about the 20 amino acid, okay? I will answer. Of course, the pure glucogenic is more than 6. I will write all of them to you in the WhatsApp group. Do you hear me? Please answer. Are you muted? Do you hear me?
Yes, doctor.
Do you understand transamination? Because I'm going to shift to oxidative deamination. All of you understand the
I haven't
transamination?
understood.
All of you
I
understand
haven't understood.
transamination?
No, no, doctor.
You don't understand
Please
transamination?
repeat. Yes, I don't.
Why? This is very easy. Trans means transport. Translocation.
Doctor, it's not easy.
It is easy. This is the amino group. I don't need noise. This is amino group. I'm going to give this amino group to keto acid, alpha ketoglutaric. The amino group is translocated, shifted to a keto acid, which is alpha ketoglutaric. And this amino group will be replaced by a ketone. So alanine will be converted into pyruvic, which is a keto acid. The amino group will be converted into C double bond O. Okay? Alanine contain amino group. We remove this amino group and replace it by C double bond O. Where does the amino group go? It goes to alpha ketoglutaric, which is converted into glutamic. Is this clear or not?
It's very clear to me.
Okay. Another amino acid, which is aspartic. Of course, any amino acid contain amino group. Any amino acid contain amino group. I will remove this amino group and replace it by ketone. The keto acid or the ketone of aspartic is oxaloacetic. So oxaloacetic is the alpha keto acid of aspartic. It has another name, carbon skeleton. Carbon skeleton of aspartic is oxaloacetic. Where does the amino group go? The amino group is translocated to ketoglutaric. The ketoglutaric accepts the amino group and it is converted into glutamic. When the ketoglutaric accepts the amino group, it is converted into glutamic. What is the benefit? The benefit is that I remove the amino group from aspartic. I get rid of the amino group. As I said, the amino group is toxic because of the nitrogen. The nitrogen is dangerous. So aspartic becomes non-toxic because aspartic loses the amino group and convert it into oxaloacetic. You know the value of removal of amino group? Okay? Is this clear? This is transamination, means exchange of the amino group, translocation of the amino group. The second is oxidation. Oxidation with deamination. Oxidation means removal of hydrogen. Deamination means the removal of the amino group. These are another enzymes, another group of enzymes. These enzymes are termed oxidases, which catalyzes oxidation. If I have this amino acid contains amino group. The first step is oxidation, removal of hydrogen. And hydrogen is carried on flavine mononucleotide, riboflavin, which is vitamin B2. And these are 2 atoms of hydrogen are removed from the amino acid. This is oxidation. Again, I don't need this compound. This compound is unstable. I don't need it. Spontaneously, ammonia will be removed. Ammonia will be removed from this derivative and it is replaced by ketonic oxygen. When I remove ammonia immediately, it is replaced by C double bond O, alpha keto acid. So this is oxidation, means the removal of hydrogen. Deamination, removal of the amino group in the form of ammonia, NH3. This enzyme is termed amino acid oxidase. This oxidase needs riboflavin, FMN. Riboflavin is vitamin B2. It accepts hydrogen. Hydrogen will meet oxygen to form peroxide. Hydrogen peroxide, H2O2. Hydrogen peroxide is converted into water by an enzyme called catalase. So do you understand oxidation with deamination? Or not yet?
Yes, Doctor.
Yes, oxidation, removal of hydrogen. Deamination, removal of ammonia. This is the ammonia, which is the amino group. What is the benefit? I convert amino acid into keto acid. A keto acid is the carbon skeleton of amino acid, and I remove the toxic nitrogen, the toxic ammonia. Another enzyme called glutamate dehydrogenase. This is also oxidation and deamination. Oxidation, removal of 2 hydrogen is oxidation from glutamic acid. It will be carried by a carrier, which is the NAD. And another step is the removal of amino group. Amino group comes out in the form of ammonia. It is replaced immediately by ketone. So glutamic is converted to alpha ketoglutaric. So I have oxidation, removal of hydrogen, deamination, removal of ammonia, which comes out as amino group. Okay? This is a combination of the 2 reactions. Trans means transamination. What is a trans? Exchange of the amino group. The amino group is exchanged, translocated to alpha ketoglutaric, and the amino acid is converted to keto acid. When alpha ketoglutaric accepts the amino group from this amino acid, it is converted into glutamic. So this amino group comes from this amino acid. So there is exchange of amino group. This amino acid gives the amino group to alpha ketoglutaric. And then alpha ketoglutaric is converted to glutamic. Again, I want to remove the amino group from glutamic because the amino group is toxic. Nitrogen is toxic by glutamate dehydrogenase. This is an oxidation because I removed hydrogen. And deamination because I removed the amino group in the form of ammonia. So this is oxidative deamination. This is actually two reactions. The first is transamination. The second is oxidative deamination. This is a combination of two reactions. The above reaction is exchange of amino group. It's called aminotransferase or transaminase. The lower reaction is glutamate dehydrogenase, oxidation and deamination. I'm not going to discuss this slide today because it will be difficult. You have to study oxidative deamination, transamination, and transdeamination. Please read it slowly from your book or from the handout, and you are going to be studied. Okay? I will answer this question from Jackson Isaac to everybody. I will answer and send you the answer on the WhatsApp group and send you the remainder of the glucogenic amino acid. Okay? Do you understand the transdeamination?
Yes, doctor.
Yes. Okay. Anyone who do not understand, please hear the video. I send you the video immediately after the meeting. So you can hear the video several times and repeat the video again until you understand. Or you read your book slowly and you are going to understand. It is not very complicated. It is simple, but you need to memorize everything you study. Okay? Thank you very much. I'm going to leave the meeting now. Anyone want to ask any question? Okay, thank you very much and goodbye.
Bye for now, doctor.
Bye. Okay, bye-bye.